Hello, good morning. This is the second part of your tutorial video on nucleic acid, which can be a consecutive video from the first part 1. This one is part 2. So when you come to part 2, this part 2 can be started by the concept of replication of DNA in prokaryotic cells. Early research on bacterial DNA replication and its enzyme helped to establish several basic properties that are proven applicable to DNA synthesis in every organism. And although the fundamental properties or features of DNA replication process and the mechanism used by the enzyme are essentially identical in all species, but extensive study were done in an Escherichia coli or E. coli, which is a gram-negative bacterium, and this prokaryotic DNA replication can have almost around 4 steps. So the first step of this DNA replication in prokaryotic can be started by a separation of the two complementary DNA strands toward a single stranded DNA, which can be used as a template for DNA replication. That it means. So to separate the two complementary DNA strands, we have to use different enzyme proteins and different things, but the first one is the DNA helicase, which can be used for unwinding the DNA double strand using an energy provided by ATP, creating the two replication forks. And the second one is the single-stranded DNA binding proteins. This protein is also called helix destabilizing protein. They are cooperatively bind only to a single-stranded DNA and they prevent the reformation of the double helix from this single strand. So they have two functions. One, it can be used for creating a single strand as well as preventing the reformation of the double helix of the DNA. So this protein keeps the two strands of DNA separated in the area of replication origin, thus providing the single strand templates required by the DNA polymerase. This DNA polymerase can be used for facilitating DNA replication in prokaryotic as well as in eukaryotic. Actually, this DNA polymerase may be different classes which can be different from eukaryotic and these prokaryotics. Yes, so this the single-stranded DNA binding protein can be used for creating a single strand with DNA helicase as well as to avoid the reformation of this double helix. So this protein keeps the two strands of DNA separated in an area of the replication origin, thus providing the single-stranded template required by the DNA polymerase. They also protect the DNA from nucleates that leave a single strand to the fragment. So this one is how DNA helicase unwinds the double helix, and the direction of the movement of replication can be toward the two forks, maybe toward the replication fork or maybe away from the replication forks. The second step is solving the problem of supercoiling. So the second step is solving the problem of supercoiling. This is also another problem of DNA replication. So as two strands of the double helix are separated, the problem is encountered, namely the appearance of positive supercoil or supertwist in the region of DNA ahead of the replication fork are there. So the accumulating positive supercoil interfere with the further unwinding of the double helix. This means that once the DNA single strand or double strand can be converted to a single strand, it can be twisted to supercoil. So this must be avoided so as to DNA replication can be takes place. So to solve this problem, there is a group of enzymes called DNA topoisomerase, which are responsible for removing the supercoil in the helix. Actually, we have different types of topoisomerase: topoisomerase 1, topoisomerase 2, topoisomerase 3, and the topoisomerase 4. So type 1 DNA topoisomerase are reversibly cut the single strand of the double helix. They have both a nuclease or strand cutting and a ligase or strand releasing activity actually. So they do not require ATP, rather store the energy from the phosphodiester bond. They cleave as well as reusing the energy to receive the strands. So this one is a double helix DNA strand, and it can be separated to a single separation or single strand, and the positive supercoil can be here obtained, but this positive supercoil can be avoided by using topoisomerase enzyme. Type 2 DNA topoisomerase can be bind tightly to the DNA double helix and make a transient breaks in both strands. So the enzymes can cause the second stretch of the DNA double helix to pass through the break and finally reseals the break. So as a result, the negative and the positive supercoil can be relieved. So again are required in both prokaryotic and eukaryotic for the separation of interlocked molecule of the DNA, and the main type of topoisomerase 2 in E. coli and the other prokaryote is called DNA gyrase. Actually DNA gyrase can be either DNA gyrase B or DNA gyrase A, and the DNA gyrase B is mainly found in E. coli, and this DNA gyrase must be inhibited by different antibiotics, especially quinoline, the one that have for example, ciprofloxacillin kill a bacteria by inhibiting a DNA gyrase which block the DNA replication. This one is the mechanism of action how antibiotic can be works by using this by inhibiting this DNA gyrase so as to avoid what blocks the DNA replication. So this shows how the protein helicase and enzyme helicase and DNA gyrase can be performed a function. So the third step is the direction of the DNA replication during elongation, and the DNA polymerase always synthesize a new DNA strand in the 5 to 3 prime direction with the 3OH as the point at which the DNA is elongated, and this means that the strand serving as a template is read from 3 to 5 direction, which is always in opposite or anti-parallel direction. Therefore, beginning with one parenteral double helix, then two newly synthesized strands must grow in opposite direction. So one is toward the replication fork, and the other is away from the replication fork. This is accomplished by the slightly different mechanism on each strand, and this strand can be known as Okazaki fragment. So this strand can be either leading strand and lagging strand, which is opposite to each other. Leading strand means a strand that is being copied in the direction of the replication fork, which can be synthesized continuously actually, and the lagging strand is a strand that is being copied away from the replication forks, and it is synthesized discontinuously with a small fragment called DNA Okazaki fragment. So Okazaki fragments are eventually joined by the ligase enzyme to become a single continuous strand. Here also RNA primer are there. The one that is very important to fill a gap during DNA polymerase cuts the strand of DNA, and this DNA polymerase cannot initiate the synthesis of complementary strand of DNA on totally a single strand template, and rather they require RNA primer, that is a short double-stranded region consisting of RNA base pair and anti-parallel to the DNA template with a free OH group on the 3 end of the RNA synthesis. In short, this RNA primer can be used for filling the gap left by the DNA polymerase or topoisomerase. So synthesized enzyme called primase synthesizing a primer where they are required. There is also a chain elongation system here. Once the replication can be started, the chain can be elongated toward different direction, that is mean. So prokaryotic and eukaryotic DNA polymerase elongate in a DNA strand by adding deoxyribonucleotide to the 3 end of the growing chain. So all deoxyribonucleotide triphosphate must be present for DNA elongation to occur. So prokaryote have 3 main type of DNA polymerase 1, DNA polymerase 2, and the DNA polymerase 3, which have different function. This one is the structure of how primer or RNA primer can be used for filling the gap left by topoisomerase by extending the chain, or chain elongation can be takes place by using RNA primer. So this one is an Okazaki, the first person who identified the 2 fragments, the leading fragment and the lagging fragment, or the leading strand or lagging strands. So what is the function of this DNA polymerase? So DNA polymerase 3, for example, catalyze the DNA chain elongation using the 3-hydroxyl group of the RNA primer as a substitute of the first deoxyribonucleotide. has very high catalytic potency, fidelity, and processivity among the three DNA polymerase, the one that have high catalytic potency, fidelity, and processivity. These catalyze the formation of many thousands of phosphodiester bonds before releasing its template that it means. Therefore, 1000 nucleotides are added per second compared with only 10 per second for DNA polymerase 1. Actually, DNA polymerase 1 have what? Low processivity. So has a proofreading mechanism for the newly synthesized DNA. If there is some mistakes during DNA replication or DNA elongation, the proofreading can be takes place by using this DNA polymerase 3. So misreading of the template sequence could result in retrace, lethal, and mutation. To ensure this replication fidelity or accuracy, DNA polymerase 3 has a proofreading activity. So in E. coli, a mistake is made only once for every 10⁻⁹ to 10⁻10 nucleotides added, and as each nucleotide is added to the chain, DNA polymerase 3 check to make certain if added a nucleotide is correctly matched with its complementary based on the template. If it is not, the 3 to 5 exonuclease activity lead to what? The mistakes activity. And this one is how the proofread function can be takes place by DNA polymerase 3. Pol1 or DNA polymerase 1 can use for removing the RNA primer and replace them by DNA, and has 3 to 5 exonuclease activities that enable it to hydrolytically remove the RNA primer. Has 3-5 polymerase activities that synthesize DNA for filling the gap left after primer can be removed. Has 3 to 5 exonuclease activity that proofread the newly synthesized DNA. So this one is a function of Pol1. The DNA polymerase 2 has a DNA repairing function. This one is, the main function of this DNA polymerase 2 has what? Proofreading function. So the final phosphodiester linkage between the 5-phosphate group on the DNA chain synthesized by DNA polymerase 3 and the 3-OH group on the chain made by DNA polymerase 1 is catalyzed by ATP-dependent DNA ligases. DNA ligase can be used for what? Joining the two fragments or strands. Here is how DNA polymerase 2 can be used for what? Joining the two fragments or two strands. So the action of DNA ligase is sealing or the gap between the adjacent DNA fragment so as to produce what? The double helix DNA structure. So the fourth step is termination of replication. So this is a place where the DNA replication becomes stopped. So replication ends when the two replication fork meet on the other side of the circular chromosome at the termination site, and the DNA replication finishes at the special site in the terminus region of the chromosome, and this termination region is the site in the terminus region that blocks the movement of the replication forks, and each terminus sequence prevents further progression of one of the replication fork when terminus binding protein is bound. So the last all about the replication of DNA for prokaryotic. Now we have to see summary here. Summary or the features of DNA replication. It can be semi-conservative, bidirectional, or two-directional. It means semi-discontinuous, catalyzed by DNA polymerase, very accurate, otherwise it can be proofread, takes place by DNA polymerase 3. The very steps of DNA replication can be either initiation, elongation, the termination step are there. We have to see for the next consecutive slide maybe. So this DNA replication process has 3 steps. Initiation, which can be recognized as starting point, which means the separate double-stranded DNA to a primer synthesis. Elongation means the double-stranded DNA to the existing strand from say phosphodiester vein, correct the mismatch base and extending the DNA strand. Termination means simply stopping water replication. This is a summary how DNA replication and the different protein as well as their function in DNA replication. For example, polymerase or DNA polymerase can be used for deoxynucleotide polymerization. Helicase can be used for unwinding of DNA. Topoisomerase can be used for relief torsional strains that result forms helical induced unwinding of the DNA. And this primase can be used for initiate the synthesis of RNA primer. and the DNA ligase can be used for sealing the single strand nick between the nascent chain of Okazaki fragment or lagging strand, that means. And the single strand binding protein can prevent the premature annealing of what? DNA, double-stranded DNA. So the next topic is all about replication of eukaryotic. EU means through this man, any organism that have a through nucleus bounded membrane. The nucleus, the membrane bounded nucleus, that is known as eukaryotic. We human beings also are eukaryotic cell actually. Have eukaryotic cells. So also this the principle of DNA replication in prokaryotes and the eukaryotes have a great deal in common. They also have significant differences. So enzyme with DNA polymerase like primase, ligase, helicase, topoisomerase activity are all present in eukaryote. Also these enzymes differ in some respects from those of the prokaryotes. So eukaryotic DNA replication is more complex in scale and it is coordinated with actually the cell cycle. Actually we have different cell cycle here, and this cell cycle may be what G1 or grows in the mechanism, or G1 phase of cell cycle, which is grows in the metabolism, and the S phase is always this eukaryotic cell cycle can be takes place in S phase of DNA replication. G2 phase, which is a preparation for cell division, and the M or mitosis phase is a place where cell division can be takes place to produce what the delta chromosomes. But eukaryotic cell cycle can be takes place among this phase on S phase of cell cycle. So what's the difference between the prokaryotic replication and the eukaryotic replication? For example, in eukaryotic, E. coli must replicate 4.8 million base pair, but human diploid cell must replicate what 6 billion base pair. How much it is very great replication can be takes place in eukaryotic, and the genetic information for E. coli is contained on a single circular chromosome, but in human being we have 23 pair of linear chromosome must be replicated. So by this way we have to differentiate these prokaryotic and eukaryotics. And also these prokaryotics cell division can occur throughout the most of the cell, but eukaryotic replication can be limited to S phase of cell cycle, as I mentioned before. So you have to see this table carefully so as to identify the difference between the two. So this eukaryotic DNA replication can also have DNA polymerase. This DNA polymerase can be DNA polymerase delta, DNA polymerase alpha, DNA polymerase gamma, DNA polymerase rhinase H, and different DNA polymerase are there here. And this DNA polymerase delta or pol delta can be major replicating enzyme that carries out a function of DNA polymerase 3 by elongating the leading strand and each Okazaki fragment. So before it act as a pol delta needs RNA DNA primer made by polymerase alpha. This polymerase alpha can be used for what? Primase synthesis actually. So it has 3 to 5 exonuclease activity to proofread the newly synthesized DNA, and this polymerase alpha is a multi-step multi-subunit enzyme, and one subunit has primase activity which initiates strand synthesis on the leading strand and at the beginning of each Okazaki fragment. So the primase subunit synthesizing a short RNA primer, and then pol alpha adds about 20 deoxyribonucleotide by its 5 to 3 polymerase activity to this RNA primer and dissociate from the template because of the low processivity of what? Pol alpha. This processivity of pol alpha is very, very low, but processivity of pol delta or DNA polymerase delta is very high as DNA polymerase 3. Then polymerase delta highly processive enzyme and adds a deoxyribonucleotide to its RNA DNA primer, continuously producing a leading strand or discontinuously producing a lagging strand. So ribonuclease H or rhinase H, an enzyme degrades and removes the RNA primer of the DNA in the RNA double helix, and the gap left can be filled by DNA polymerase delta, and as in bacteria, DNA ligase is subsequently used for joining what? The Okazaki fragments. DNA polymerase gamma is mainly found in mitochondria and which can be used for replicate mitochondrial DNA. So the remaining polymerase are involved in repair of damaged cell or DNA, and the eukaryotic replication occurs with high fidelity, approximately 1 mispairing for 10⁻⁹ to 10⁻12 nucleotide. This one is the summary how different DNA polymerase can be used for eukaryotic DNA replication, and these all enzyme DNA polymerase can have different functions, so you have to see in the read for further reference so as to build your knowledge on this DNA polymerase. So this one is a direction of replication fork. Always I mentioned before, this replication can be takes place either towards the replication fork, which is leading strand, or away from the replication forks that is known as lagging phases. So this is the way how the replication can be takes place, and chain extension can be or elongation can be takes place by using different polymerase and different proteins. Here is a major protein involved in the replication of eukaryotics. As I mentioned before, DNA polymerase, trimase, helicase, single-strand proteins, topoisomerase, and the DNA ligase all can be in the same fashion involved in the replication of eukaryotic as that of the prokaryotics. So, what would happen in eukaryotic DNA replication? There is one problem. That problem is known as the end replication problem, and this problem can be solved by maybe telomers. So let's see what is the problem and how this problem can be created. So eukaryotic cell faces a special problem in replication, the end of their linear DNA molecule, or end replication problem. As the DNA replication approaches the end of the chromosome, the problem develops in the lagging strand actually. So either primase cannot lay down the primer at the very end of the chromosome or after the DNA replication is complete. Therefore, the RNA at the end of the chromosome is degraded following the removal of the RNA primer from the extreme 5 end of the lagging strand. There is no way to fill the remaining gap with the DNA. Consequently, the newly synthesized strand is shorter at the 5 end, and there is 3 overhanging in the replication of the DNA strand. So if the chromosome becomes shorter with each other, successive replication and genes would be lost. So circular prokaryotic DNA molecules do not have ends, and so do not have this problem. And to minimize the deletion of important gene region, the chromosome end are formed by telomers. Therefore, this end replication problem, which can be associated only on the eukaryotic DNA replication, can be solved by these telomers. This is by lengthening the 3' overhang by the addition of telomers, so that the primase can bind and synthesize a complementary strand. Telomerase can be an enzyme that is used for making these telomers. And as you see in the diagram, the RNA primer at the 3' position can be left a gap. So this gap can be filled by telomers. Otherwise, it leads to the problem of end replication problems. So the telomers are a double-stranded non-coding G-less deoxyribonucleotide repeat at each end of the eukaryotic chromosomes with associated proteins. Telomers consist of repetitive sequence of thymine and guanosine, and the thymine-guanosine strand is longer in its complement, leaving the region of single-strand DNA at the 3 end of the double helix that is a few hundred nucleotide long. So the single-stranded region of the fold back to itself is forming the structure that is stabilized by protein. This complex protects the end of the chromosomes. So it is these telomers that can be used for filling the gap and making the end replication problem become solved. So the enzyme telomerase contains both proteins and RNA and act as an RNA-dependent DNA polymerase, just like the reverse transcriptase. The RNA within telomerase contains a complementary copy of the repeating sequence in the telomers and can base pair with the existing 3 overhangs. Therefore, the polymerase activity of telomerase will use the existing 3-hydroxyl group of the overhang as a primer, and it is own RNA as a template, and synthesize a new DNA that lengthens the 3 end of the DNA strand. So the 3 overhang can also form the complicated structure with telomerase binding protein to protect the end of the chromosome from damage and the nucleus attack once they have been lengthened. So telomerase loop to seal the end of the chromosome, and the organism seems dependent in part on progressive oxidation damage to long-lived macromolecules, and the replicative cell sensing seems to be caused by change in the structure of the telomerase, and because many human somatic cells are telomerase deficient, their telomers shorten with every cell cycle division, and their protective proteins cut progressively. So eventually the exposed chromosome end are sensed DNA damage, which activates what p53 dependent cell cycle arrest. This p53 chromosomal enzymes that found in our body is very important in inhibiting or combating proliferation of cells and protecting our body from cancerous cells. Unfortunately, telomerase do not grow as we grow, and we find the telomerase activated only in a gamete, in stem cells, and in most cancer cells, they have the ability to produce telomerase. So as a result, they do not undergo replicative cells, and if cancer cell would rapidly run out of telomerase, they would be die off. So telomerase inhibitors are the potential anti-cancer drug because they stop the tumor cell from replicating over and over again. So this telomeric shortening has been linked to the genomic instability, cell aging, and this actually, as our age can be increased, this telomeric length can become decreased. As you see from this diagram, as age increases, length of telomere become decrease. So the function of telomere in human, in short, can be what protect the chromosomal end from recombination, end-to-end fusion, and the recognition as a damaged DNA, enables the complete replication of DNA, enhances genomic stability, DNA repair by preventing degradative action of exonuclease, participate in regulation of gene expression, and serve as a mitotic lock which shorten with each cell division. So this all is about, in short, today we have to see what about DNA replication. This DNA replication can be classified as eukaryotic and prokaryotic replication, which are slightly different actually, and we have to see different enzyme, proteins, and different activators can be used in both cases, and the function are almost similar, but we only slightly difference are there. So you have to try to identify the differences, and in the eukaryotic replication especially you have to know the concept of end replication problem, which can be solved by the telomeres, and these telomeres lengths can be decreased as we are aging, and that can be not only for this filling the gap, can also be used for what inhibiting cancerous cell in our body by producing telomerase enzyme. So this is all about today's tutorial video. So until the next tutorial video on part 3, thank you for watching, and don't forget to subscribe to the channel and share link and comment, as well as like the channels, so as to get what the hot and important tutorial video for you. Thank you very much.